Yeah, ma'am, it is on live actually. You can start the program.
Good morning to you all, and greetings on behalf of the Department of Chemistry, PV Siddhartha College of Arts and Science. Respected principal of our college, Dr. M. Ramesh Garu, Professor Lagada, Director 3D Lagada Garu, and today's episode we talk research scientist, Vijayawada, discovery, all the learners, faculty members, and students from various colleges, universities, and alumni of our department. We learn about discovery. This topic is very much the need of the hour. All of us are very aware of the term antibiotics and their invaluable role, not only in the past, but also in the pandemic. Antibiotic resistance affects both global health, food security, and development goals. This resistance any country is this resistance is rising to an alarming high level in all parts of the world and also the major concern of the World Health Organization. The world urgently needs change of pesticides and antibiotics. Without urgent action, we are heading for post-antibiotic era, which even common infections can kill human beings. And also COVID-19 provides a stark reminder to all of us to be prepared in advance before a disaster hits. I am glad that our resource person is addressing us on most important and relevant topic. I hope this will be a fruitful and meaningful time for all. Thank you. And now I request our Principal Sir, Dr. Ramesh, to greet all the participants of
Good morning to all. I welcome Dr. Venkatesh Lagada, research scientist, who is our alumnus. Really, we are proud of you, sir. Well, as we are working as a research scientist in Canada and representing our college in Canada also. So we are so proud of you. The present days, there is a shortage of scientists in all the areas, not only in the chemical chemistry, in physics and biological sciences, in each and every area, there is a shortage of so-called scientists. So hopefully you have chosen the right choice, you have taken the right choice and to grow and reach the greater heights also, I think. So I thank, first of all, for accepting the invitation from the Department of Chemistry for delivering a lecture on the antibiotic discovery strategies in the era of drug resistance. So actually, this is a right topic to the present days. So nowadays, so many people are using the antibiotics in overdoses and some complications, different types of complications they are facing also. So a right use of the antibiotics is also very important. The people like you can suggest all the people to use the antibiotics in a right way, and they can cure out of them, come out of these various types of the diseases, whatever it is. So I advise all the participants to make use of this session, very fruitful, and also I congratulate the Department of Chemistry for arranging of this type of the wonderful session today. Thank you. Thank you very much.
Yeah, good morning, everybody. This is Rajesh Jampala, Dean, PV Siddhartha College of Arts and Science. And good night to Venkatesh Lagada at Canada. I know that it is a very odd time for you to take up this session. Thanks for accepting our request on the week. I mean, in that sense, actually, we are proud of you, as our principal said just now. The college is represented at Canada at the highest level is always a very heartening news for all of us. We seek your support in strengthening our chemistry department in collaborative efforts. I also request you to help our students also to get settled in scientific research or advancement in that particular area. As our principal and our HOD said, this is a time where most of the countries are facing problems with pandemic or endemic situations in COVID. And the topic is also relating with antibiotics and the side effects of such aspects. Collaboration is a key word here. One country cannot solve all the problems of the mankind. At the same time, one institution is not in a position to handle with great depth. So in that sense, we seek your support in strengthening your alumnus, PV Siddhartha College of Arts and Science, to have a collaborative projects in future. And I congratulate the Department of Chemistry for organizing this particular event. At the moment, Vijayawada is observing as rainfall actually. And today the entire state is following the Bandh also, but still we are in the process of learning something from you. We are proud of you, Venkateswarlu, and I once again congratulate you for holding that particular position of research scientist level 2 in medical chemistry and drug discovery, Dalredia Drug Discovery Incorporation, Toronto. Wish you all the best, and thank you very much for accepting our request.
Madam, your voice is not audible.
I think
Thank
David
you
Raju
very much, sir. Now I request Dr. M. David Raju garu to introduce our today's resource person.
Good morning to one and all. It's my privilege to introduce Dr. Venkateswarlu Lagada. We proudly say that he is our old student only. Currently, he is working as a research scientist too at Lagada Drug Discovery Inc. in Toronto, Canada. After completing his PhD in November 2015 at Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore, India, in medicinal chemistry and antibiotic drug discovery, he joined at McMaster University, Canada as a postdoctoral research fellow. His research work has been mainly focused on addressing the ever-increasing antimicrobial resistance by employing the chemical and synthetic biology strategies. His research work has been published in 24 international peer-reviewed research articles and currently having an H-index of 17 with greater than 1,250 citations. Further, he has been a co-inventor of various national and international patent applications from 4 different inventions. 2 inventions have been granted in different nations: India, USA, Canada, Europe, Australia, South Korea, and a license to Viprajan Biosciences, India. He has been awarded the prestigious Gandhi Young Technology Invention Award, India, in association with the Biotechnology Industry Research Assistance Council in 2015. He was also the recipient of 2 gold medals in undergraduate studies and received 2 Best Research Poster Presentation Awards at the international conferences recently. He has received Michael G. D. Groot Fellowship Award to pursue postdoctoral research at McMaster University, Canada. His research interests include development of various simple chemical and synthetic biological strategies to discover new class of durable antimicrobial and the role of antimicrobial in the disease progression of Alzheimer's. Awards and recognitions. In 2016, Michael G. D. Groot Postdoctoral Research Award, McMaster University, Canada. In 2015, Gandhi Young Technological Innovation Award. In 2014, International Travel Grant from Science and Engineering Research Board, Department of Science and Technology, India. In 2013, Best Poster Presentation Award at CTDDR 2013. In 2012, Senior Research Fellowship, Council of Scientific and Industrial Research, India. And Best Poster Presentation Award at Mid Chemical Research Society of India meeting, and etc. And a research grant, Biotechnology Industry Research Assistance Council, Gandhi Young Technology Innovation Award in 2015, received this grant from Society for Research and Invitees for Suitable Technologies and Institutions in association with BIRAC as a principal applicant during PhD and received rupees 15 lakhs for the project development. And reviewing, he is a reviewer to many articles, and that's all. Thank you so much.
Now I request our today's Thank you very much, Dr. David Raju. Now I request our today's resource person, Dr. Venkateswarlu Yarlagada, to take over the session.
Yeah, so can everyone see my slides? Can you see my slides? Can you hear me? You can see me? You can see my slides? Okay. You cannot see. Can you see my slides? Oh God. Can you hear me? Oh, shit. Okay, can you see my slides? Oh, this is going to be difficult. I don't know why I'm. Hello? Can you see my slides, ma'am? Not. I don't know. I'm sharing the screen. Okay. All right. Yeah. Okay. Okay, ma'am. Hello, everyone. Good morning. So thank you very much for the kind introduction. So today I'm going to be talking to you about some of the antibiotic drug discovery strategies in the era of drug resistance. So the reason I say in the era of drug resistance, probably you would understand in the future slides. So why I call right now we are in the era of drug resistance. So the title of my talk, as I said, the content of the talk is been divided into 4 sections. So basically, initially, I would like to introduce 2 infectious diseases, and then followed by antibiotics and their importance, and then further I would talk about bacterial resistance to antibiotics, and then I would talk about some of the strategies that we developed to overcome this antibiotic resistance for the past decade. So being talking about infectious diseases, so today I would like to introduce you like what are the infectious disease causing agents. So basically like bacteria, virus, fungi, and parasites, these are the organisms that cause infections. So here I want to stress a point that so antibiotics act only against bacteria, and antivirals against only virus infections, and antifungus only for the fungi. And I like that, like parasites, for parasites only antiparasitics. So basically if you if you have any other infection, if you want to take any other drug other than antibiotic, or other than if you have a bacterium infection, you have to take only the antibiotic. If you have viral, only antiviral. So if you take a cocktail of drugs, that means you are actually wasting the the drug arsenal. So here I would like to introduce you the endemic and pandemic. So the endemic is the one, like for example, if you take an like 2 years back in Kerala, we have a Nipah virus endemic. So basically, so that Nipah virus has appeared for only some special geographic region, only in Kerala for one district, which is called endemic. But if in case of pandemic that we see COVID-19, right? So it crossed across the world and throughout the geographical region, so which made it like a pandemic. So so here I would like to show you some of the timeline of infectious diseases. So so on the left hand side, you see that the number of people died over the time, and on the right hand side you see the number of pandemics that appeared for the past 2000 years. So here I show you that like some of the pandemics that are caused by the bacteria. So that I showed you in the red boxes, and then I'm showing you some of the pandemics that are caused by the virus, like Spanish flu, Russian flu, HIV, and now COVID-19. So these are all happened at the beginning of 20th century. So if you carefully see the timeline of these infections and pandemics, so basically until the 20th century, you see most of the pandemics are caused by the bacteria. And then from the beginning of the 20th century to now, we see mostly the viral pandemics, right? So basically during this region, like from 1910 to now, we have the wealth of antibiotics. So the antibiotics were discovered during that phase. So we have the antibiotics during this period. So hence, so we were actually able to treat the bacterial infections during this region, this timeline. But in the other cases, like where you see before the discovery of antibiotics, so there were no antibiotics to treat. So hence there were many bacterial pandemics. So so here I would like to show you that. So what happened to the world with no antibiotics? So how did the doctors treat infections, right? So during the like in case of Asian countries, we have like Ayurvedic medicine or natural therapies. But in case of Western countries, like so there used to be a bloodletting experiment. So so they think that so those infections were thought to be caused by the excess of blood. So they used to let the blood from the body, from different parts of the body, like I show here. So basically they thought that like if depending on the symptom and the kind of infection, so they think that like if you let the blood from the body, you can clear from the infection. So there used to be barber surgeons. So they use knives and leeches to let the blood from the body to clear the infection. So this treatment worked for some people, but did not go well for many people. So basically, for example, here George Washington, so the president of American States, so he actually so he got a throat infection caused by Streptococcus pneumoniae. So he was so he was asked to let the blood to clear from the infection. So what he saw is so for about the doctor actually let his blood about 4 liters in a period of 10 hours. So as a result, so because of the lots of blood he lost, and then he eventually died because of the too much of blood removal. So having said that, so don't try bloodletting yourself if you have an infection. So now we are fortunate to have antibiotics. So being talking about antibiotics, so the antibiotics were first discovered by Alexander Fleming, right? So basically the Alexander Fleming was a microbiologist. So he was growing a Staphylococcus aureus bacteria on a plate. So he went for a vacation for 2 days. And then what happened was, so when he was growing the bacteria on a plate, so you see he got a contamination on the same bacteria plate. So and also you also see that here around the fungal contamination, you see that there is no bacterial growth, but you see the growth away from the fungus, right? So he thought that maybe the fungus is producing something that is inhibiting the growth of the bacteria, and he eventually went to purify the compound from this fungus, Penicillium notatum, and he identified to be penicillins, which are the first natural antibiotics to kill the bacterial infections. And then he also said that, I want to make a stress point here. So it is not difficult to make the microbes resistant to penicillin in the laboratory by exposing them to concentrations not sufficient to kill them. So he was saying that if you expose the bacteria to penicillin at sub-MIC level, at sub-inhibitory concentrations, so the microbe can get resistant to the penicillin, and penicillins may not be no longer active. So which we call now antibiotic resistant. So here is the discovery of antibiotic pipeline. So basically, most of our antibiotics have come from the natural origin. Like so the one that I showed you in the black in color, so they came from the nature, and then the one I showed you in the maroon color, they came from the synthetic origin, like sulfonamides, quinolones, and oxazolidinones. So basically, most of our antibiotics have come from the natural origin. So here I would like to show you the importance of antibiotics in the history of human medicine. So basically, the life expectancy of a human being before the discovery of antibiotics was 47 years. And then with the discovery of antibiotics, so if you see the global population here, right? So initially, before the discovery of antibiotics, you see that like a stable, like a straight line. So there is no growth in the population, not much. And then here also not very much, but here where we have the antibiotic discovery occurred, so there you see that exponential growth. So basically those antibiotics played a huge role in the history of human medicine. And the same thing you can observe that, so the life expectancy of a human being after the discovery of antibiotics was about 80 years. It's almost like 32 years increased. So these antibiotics, what they do, so basically they act on the so they inhibit the essential cellular process in bacteria. So the bacteria mainly classified into two categories, like gram-positive and gram-negative bacteria. So in the case of gram-positive bacteria, we have the cell wall. So basically what these antibiotics do are, so basically they inhibit the essential cellular process like protein biosynthesis, cell wall biosynthesis, RNA synthesis, DNA, outer membrane, folic acid, and all these essential processes. And then they kill the bacteria. So bacteria acquired resistance to antibiotics, right? As I said, like, so they can acquire resistance. How they acquire? So basically, when the antibiotic enter into the cell, so the bacteria can produce enzymes which are called antibiotic degrading enzymes. So they can degrade the antibiotic, and then it makes no longer active. And then further, so once the antibiotic enters the cell, so they can efflux. So they throw out from the cell antibiotic, and also they can modify the target. So like that, so they acquire the resistance. And also there is something called intrinsic resistance. They are naturally resistant to antibiotics because of the lack of the antibiotic target, or the antibiotic is not able to penetrate through the cell. And then so here I show you like how generally they acquire resistance in a population of a bacteria. So for example, if you take 1 million bacterial cells, and then if you think all the bacteria are sensitive, but like say if you take one bacterium was resistant out of 1 million, and then that one bacterium will be survived after treatment with antibiotic, and then that resistant bacterium would be carried over further, and then they will be propagated, and they can be produced more and more, and then they become drug resistant. And then so for making the drug resistant, so they have the genes in them, right? So those genes are actually transferred to within the species or interspecies, and then they get the so like that, so the resistance is being transferred from the between the species also. And then there is another thing called bacterial biofilms. So here what they do is, like, the bacteria, they attach on a surface. Like, for example, if you take a catheter, or if you have a teeth catheter, something like that. So basically on those catheters, so the bacteria attach on the surface, and then so basically these bacteria was initially sensitive, but when they attach on a surface, they make a layer by layer by assembly, and then once they make a layer by layer by assembly, so they produce a matrix they called polysaccharide chemicals. So those chemicals, they protect the bacteria. So they protect the bacteria from the external stress. So basically, so the bacteria now here are like sleeper cells. So they hide within the polymatrix, right? Even though the bacteria is sensitive, but even if you treat with an antibiotic, the antibiotic is not able to penetrate through the polymatrix, and hence the antibiotic is not active, which we call as antibiotic tolerance or phenotypic resistance, right? So so here I would like to show you a brief statistic, like how many deaths are happening because of this antibiotic resistance. So here I'm taking an example with respect to United States only, so which is a very developing country, right? Developed country. So in a country like that, so they have about 2 million people infections happens every year, and then out of 2 million people, there about 23,000 people die every year. And then so there is a statistics that, like, if we do not tackle this resistance now, so there could be 10 million deaths by 2050. So in other words, a person can die in every 3 seconds if we don't tackle now this resistance, antibiotic resistance. So and here I would like to show you why this is because of, like, this antibiotic resistance over the time, like it is increasing, and then the discovery of antibiotics is declining over the time. So we need new antibiotics. We need durable antimicrobials to cure such infections, to have the to have that the like drug arsenal to our medicine. So during my PhD and postdoc, I was involved in the discovery of antibiotics. So broadly in 2 categories, like antibiotic adjuvants and novel antibacterial candidates. So in the case of antibiotic adjuvants, so basically these the adjuvants are the compounds. They are not active by themselves, but they are active when they're in combination. So they target the resistance mechanism, or by enhancing the activity through indirect mechanism. So in the case of novel antibiotic candidates, we have the novel scaffolds, or semi-synthetic analogues, nature product drug discovery, genomics driven drug discovery, and nano antibiotics. So today I would like to talk to you some of these strategies that we developed during the past decade. So initially I would talk about some of the semi-synthetic analogues that we developed during my PhD. So here we choose a drug called vancomycin. So the vancomycin is a glycopeptide antibiotic. So this has a glycan sugar moiety, and it has a peptide moiety, which we call the glycopeptide. So this antibiotic, what it does is, so it inhibits the cell wall biosynthesis in bacteria. So here I take an analogy to the normal compound wall. So in the case of compound wall, we have the building blocks in the form of bricks, right? So in the case of bacteria, we have those building blocks in like NAMNAG sugar residues. They have the glucosamine and muramic acid sugar residues, and those sugar residues, they have the pentapeptide, and that pentapeptide. So these building blocks. So in the case of compound wall, we have the bricks and also the cement, right? So the cement is needed to get the strength to the wall, right? So here for this, in the case of cell wall of the bacteria, we need the cementing step to get the strength to the bacteria. So that cementing step is happening through the transglycosylation and transpeptidation bond, that cross-linking. So that cross-linking is necessary for the bacteria, to get to get protected from the stress. So what vancomycin does is, vancomycin binds to the D-ALA through 5 hydrogen bonds. So because of the D-alanine binding here, that cross-linking between the pentapeptides did not happen, and hence bacteria would die because the cell wall weakens. So bacteria acquired resistance to vancomycin by modifying the last amino acid from D-alanine to D-lactate, right? So here D-alanine and then here D-lactate. So as a result, you see that so that single atom exchange from the alanine to lactate, you see the presence of lone pair repulsions and also the lack of one hydrogen bond. And because of that, so the drug would not be able to bind more efficiently with the target peptide, and hence the activity will go down by 1,000-fold. So we thought that can we improve this lost binding affinity to the vancomycin by doing some simple synthetic methodology? So with that rationale, we have come up with this article where the authors have co-crystallized the vancomycin with the lysine DLR with the tripeptide. As I mentioned in the previous slide, so the vancomycin binds to DLR through 5 hydrogen bonds, and vancomycin also makes a 6th hydrogen bond through a bridging water molecule, so which was not shown before. So basically this bridging hydrogen bond happens with the third amino acid, the lysine, right? So here our idea was like if we can extend the backbone of vancomycin that has only the until carboxylic acid further with some of the hydrogen bonding able moieties such as sugar moieties. So those sugar moieties carry the hydroxyl groups, and those hydroxyl groups can act as both hydrogen bonding donors and acceptors, right? So those moieties can have the additional hydrogen bonding affinities with the rest of the pentapeptide, and hence we can improve the overall binding affinity. So with that rationale, we have synthesized several vancomycin sugar conjugates varying the sugar moiety from the cyclic glucose, galactose, cellobiose, maltose, lactobionic acid, and acyclic glucose. So basically here, as you see that, we also vary the linker here, like from the C to oxyethylene linker and a propylene linker with a secondary amine, and we have a propylene linker with a amide bond, and we have the cyclic and acyclic in the same moiety. So after synthesizing these moieties, we check their binding affinities. So as I mentioned, so the vancomycin to the resistant peptide having the very low binding affinity, so in the order of 2. And we need about 750 micromolar to kill the bacteria for vancomycin molecule. But after synthesizing these molecules, we check their binding affinities. What we identify was that so the binding affinity is gradually improved with the addition of sugar moiety, and then the molecule that has the amide bond in the linker having the binding affinity in the order of 4, which is almost like 100-fold more affinity towards the target peptide. So, and also the activity, as you see here, in the case of vancomycin, as I said, you need 750 micromolar. But like after conjugating this lactobionic acid to the vancomycin, we see the activity at 36 micromolar. So it's almost like 30-fold more activity than vancomycin. And further, so the value, what we achieved here is not very good, but to increase the activity further, so we choose the optimized sugar conjugate. And then to this optimized sugar conjugate, we added the lipophilic moieties, so some alkyl chains to the vancomycin. So after adding the lipophilic moiety to this optimized sugar conjugate, we see the activity number 0.7 micromolar, right, which is almost like 1,000-fold more active than vancomycin. So to show you how we synthesize these molecules, I just want to show you a brief slide, like how we make. So we do like multi-step synthesis of to make these molecules. So at each and every stage, we do a glass column chromatography for the intermediates to get the pure molecule. And then once we get the pure molecule at the intermediate stage, and then for the final molecules, for the final drug candidates, so we do HPLC to purify those molecules, reverse phase HPLC. And then once we get the pure molecule, and then we do a series of characteristic studies like NMR, HRMS to get the to get the structure of the compound, what we need to get it. And then so here I want to show you like how the chromatogram looks like in HPLC and the mass spec. So to just show you like how the purity of this compound. So we need about 95% purity to check for the biological activity studies. So once we after we find the activity in vitro, as we identify Van-LBS-C10, like vancomycin having the lactobionic acid moiety sugar, and then has the C10 long chain. So having that moiety is the optimum candidate from this strategy. And then we check the activity in the mouse infection models. So basically here what we do is we infect the mice with the bacteria and then treat with our candidates to see if the compound having any activity in the mouse infection models. So here we choose two bacteria, Staphylococcus aureus infection, which is a tissue infection, and then the kidney infection. So with enterococci infection. So after treating with the antibiotic, we noticed that. So here we use vancomycin and linezolid as the control compounds for the to compare this with our drug molecule. So here this red arrow indicates that is the bacterial concentration. We infect the mice, and then in the case of no treatment, you see that the bacteria has outgrown to the 10 logs. 10 logs see a few colony forming units. And then in the case of vancomycin and linezolid, you see like a slight reduction in the bacterial count. Whereas in the case of Van-LBS-C10, it reduces so much compared to vancomycin and linezolid. And the same thing can be seen in the case of kidney infection as well. So indicating that this compound showing the better activity than those comparator drugs. And then further, we have also done the thorough pharmacokinetics and pharmacodynamic studies. So I'm not going to be talking to you today about those pharmacological studies today. But I would like to show you, I would like to tell you the comparison, like, so about 20, so this compound is about 20-fold more active than vancomycin when we compare with the ED50 value. And further, this compound was not having any toxicity. And then now on, I would like to move on to the second strategy that we developed. So basically, as I said, so the vancomycin inhibits the cell wall biosynthesis, right? So here our idea was to incorporate a new mode of action to vancomycin, or additional mode of action to vancomycin, right? So the cell wall is followed by cytoplasmic cell membrane in bacteria. So these cytoplasmic cell membrane, so these, whatever I'm showing here in the pink heads, and then these long chains, so those are the phospholipids. So here I'm showing the structures of those phospholipids, right? So those phospholipids are like lipophilic, are hydrophobic, and anionic in nature. So here our idea was if we can incorporate a lipophilic and a cationic moiety to vancomycin through the carboxylic acid, right? So that lipophilic cationic moieties can have the electrostatic, because of the cationic and anionic interactions, it can have the electrostatic interactions. And then this vancomycin part would be acting at the cell wall region, so that we can have a second mode of action to vancomycin other than the cell wall biosynthesis inhibition. So with that rationale, we have synthesized a lipophilic cationic vancomycin analogues by varying the lipophilicity from the ethyl to octadecyl. So for making these molecules, so we like I said before, like at each and every intermediate, like we purify them by glass column chromatography, and then the final molecules we purify by HPLC, and we identify the structure by NMR and HRMS. So once we identify the structure and once we have the desired purity of the compound, then we do a series of studies in the microbiological studies. So here, as we expected, so here I would like to just show you here. So as I said, the cell membrane is made up of like phospholipids, right? So those phospholipids are, as I said, like anionic and lipophilic in nature. So and also we have the potassium and also the bacterial cells, they contain potassium and sodium ions, right? So if we have a cationic moiety in vancomycin and lipophilic moiety in vancomycin, so those cationic and lipophilic moieties would be interacting with the cell membrane through the phospholipids, and then because of this charge interactions, so there is an imbalance between the potassium and sodium ions going out and coming in. So there is imbalance, and as a result, you see the potential across the cell membrane is disrupted. That's what we see here. So here, in case of here we measure the membrane depolarization. So with a dye called DISC3 dye. So basically, what it does is, so it measures the potential of the bacterial cell membrane. So in the case of if you see the increase in fluorescence intensity, that means that those compounds are causing membrane depolarization. So that's what we see here. Like in the case of VAN-QCI, that's carrying the C8 long chain and C14 long chain, you see the increase in the fluorescence intensity, indicating that these molecules are actually depolarizing the bacterial cell membrane. And further, they also go into the cell, and then they target the unknown targets. And further, these compounds also inhibit the cell wall biosynthesis greater than vancomycin. So having said that, so yes, so these molecules are having a new mode of action than vancomycin, right? So here and then we check the activity of these compounds against various bacteria. But here I'm showing you a few bacterium here. So so here we choose like vancomycin resistant enterococci VRG and Staphylococcus aureus VRSA and MRSA. So these bugs. So if you see here that like in the case of VanQ C14 and C8 that carry C14 long chain and C8 long chain, so that is the optimum chain length to have the optimum activity. And if you see the activity here for vancomycin, as I mentioned in the previous slide, to kill this bacteria, we need about 750 micromolar. And here for this compound, we need about 0.7 micromolar, indicating that these compounds are about 1,000-fold more active than vancomycin. And here there is an interesting study that so here we wanted to expose, so we wanted to see whether these compounds trigger the resistance in bacteria if we treat for a long time, like we see the antibiotic resistance development, right? So here, so basically we serially expose the bacteria to these compounds for about 55 passages, and see the MIC, see the activity of these compounds from the day 1 to day 55, and see if there is any resistance occurrence for these compounds. And what we noticed that like the MIC, the activity of the for these compounds from day 1 to day 55 remains to be the same. But whereas for the parent drug vancomycin, we see the bacteria acquire resistance to the parent drug, indicating that so bacteria face a difficulty in acquiring resistance to these compounds. And then we have also checked the activity of this the optimizer candidate VanQC, right, in the kidney infection and tissue infection model, and without any surprise, so this compound also showed very good activity in both the infection models. Like as you see here, vancomycin and linezolid, they were like mild activity for both the infection models, but VanQC had showed a very good activity. And then so here also we did a thorough pharmacokinetics and pharmacodynamic studies, but here I would like to show you a summary slide of that. So this VanQC was about 30-fold more active than vancomycin when you compare at the ED50 value. And then this compound is having the improved pharmacokinetic properties, like this compound can stay in the body for a longer time in the mice, and then having no toxicity up to 100 milligram per kg. And further, as I showed you, these compounds are having like the bacteria face the real difficulty to acquire resistance to these compounds. Then having shown that, having the sugar conjugate in vancomycin, so can have the improved binding affinity towards the target peptides, and then having the lipophilic moiety, cation moiety, can have a new mode of action to vancomycin, right? So our rationale was next, like, so to have both the properties in a single molecule, single system, so that we can effectively tackle this bacteria more efficiently, right? So with that motivation, we checked, we synthesized several lipophilic, cationic, vancomycin, and sugar conjugates. So here also we varied like the lipophilicity from the hexyl to tetradecyl. Once we synthesized these molecules for the desired purity, with the desired purity and with the desired structure, so we checked their activities. So here I'm not showing the activity for all the compounds, but I'm just showing the activity for one compound, which is the optimizer candidate. So here, so as I mentioned in the previous slide, for VRE bacteria, for vancomycins, you need 750 micromolar. I'm stressing here. And in the case of Van-LBA, which is so the compound that has only the improved binding affinity is having the activity 36 micromolar, which is about 30-fold more active than vancomycin. And the one that has the membrane active properties, Van-PYC8, so that has the activity at 3 micromolar, which is about 280-fold more active than vancomycin. But the compound that has both improved binding affinity and the membrane active properties, the compound is having a 0.09 micromolar, right? So which is about 8,000-fold more active than vancomycin, which is the very good activity. And then we have compared the activity of this compound, this optimizer candidate, with the recently FDA approved drugs, right? So as you all know, the FDA is the drug approval center where it approves the drugs before it's to the clinics. So recently these drugs like dalbavancin, chalavansin, voltamancin were approved to treat such infections. And these compounds, so this Van-PYC8-LBA, this compound was more active than these three comparator drugs. And then, as I said, so this compound was designed to incorporate all the properties, and as expected, we see the membrane active properties by improved binding affinity and improved cell wall inhibition. And then we checked the activity of this compound in the kidney infection and the tissue infection, and this VAN-PY-CAT-LBA showed a very good activity in both the infection models, like both the VRE infection model and Staphylococcus aureus tissue infection model. And then we checked the so here I would like to show you a slide for this compound, the pharmacodynamics and pharmacokinetics. So basically, pharmacodynamics is nothing but so you are varying the dose of a compound at different doses, and then seeing the activity of the compound in the mice body. So basically, so this compound, this VAN-PY-CAT-LBA, it can show the activity at 1 milligram per kg, as you see here. So it shows and it shows a very good dose-dependent activity study. And then in the case of single dose pharmacokinetics, what we do is, so we measure like how so basically we measure like how long the drug is staying in the body, right? That's called pharmacokinetics, like how long the drug is staying in the body, and at what intervals we need to take the second dose. So here for measuring that, what we do is we inject the antibiotic to the mice body, and then after we inject the antibiotic into the mice body, so we collect the blood at different time intervals, and then after collecting the blood, so we isolate the plasma, and then that plasma we process to determine the concentration of the drug molecule in the plasma at that particular time point, and then by HPLC or LCMS. And then once we identify the compound concentration, we do a curve analysis. And then once we do a curve analysis, so we get the half-life, like how long the drug staying in the body. So the half-life of this VAN-PY-CAT-LBA compound was about 5 times more than the parent drug, indicating that this compound also having the improved half-life or improved pharmacokinetic parameters compared to vancomycin. And then the compound was also not toxic as expected. So here I would like to compare. I would like to take a pharmacodynamic point called EDU2 log kill. So basically, ED2 log kill, as I said, like the pharmacodynamics is nothing but like you are doing a dose-dependent study. So basically, so in the case of pharmacodynamics, so the parameters like ED50, stasis, or ED1 log kill or 2 log kill. So here if you see, so here is the concentration of the drug molecule on the x-axis, and the y-axis you see the bacterial count, right? So here, so the dose that is required to kill 2 logs of reduction from the initial count is called ED2 log kill. So to achieve that pharmacodynamic point, so for the drugs like linezolid, vancomycin, and telavancin, you need about like 500 milligram per kg, 288, 80 milligram per kg to achieve that pharmacodynamic point against this particular bacteria. And then in the case of like these 3 compounds that we developed in the strategy 1, 2, 3, so these 3 compounds were more active. As you see, like you need about only 11.2, 8.8, and 5.8 milligram per kg. So which is like way more active than these 3 compounds. And then if I compare within these strategies, so as expected, so the compound that having the membrane active property and improved binding affinity that having the more activity, if I compare within these strategies. So then I would like to show you a completely different strategy of semisynthetic strategy. So as I said, like in the case of cell wall, right? So the cell wall is, as I said, like it has the building blocks like NAM and NAG sugar residues with a pentapeptide. So these precursors actually they're being synthesized in the cytoplasm. So basically, when they're synthesized in the cytoplasm, they have to be transferred to the periplasm. So for transferring those precursors, you need a lipid recycling molecule called C55 pyrophosphate. So this pyrophosphate is necessary to transfer this precursor to the periplasm. So basically, the idea was, can we target this lipid pyrophosphates, the cell, and also this has to be free again to recycle them. So can we target these pyrophosphates with the use of vancomycin molecule? So the idea was like, so it is known in the literature that there is something called dipicolyl moiety that I showed in the here. So the red color. So that moiety. Basically this compound, this ligand having the very high affinity towards the zinc 2 plus ions. So when it is bound to the zinc 2 plus, that whole complex having the very high affinity towards the pyrophosphates. So when they have the pyrophosphate binding affinity, so this vancomycin part would be acting at the DLR here, a DLR lactate, and then this moiety would be acting at the pyrophosphates. So hence now we are we are stopping both the transpeptidation and as well as the lipid recycling into the periplasm. So that was the idea that we thought, that we envisioned, and then to check, we just synthesized just one molecule to see our hypothesis is working or not. So so beautifully, so this compound, like, so yeah, so this is how we make this molecule. So we purify this molecule, as I said, like by HPLC and NMR, and we characterize by NMR and mass spec. So once we get the desired purity, we check the activity. So with that, like that single molecule, as which we call as dipivan, right? So as I mentioned in the previous slide, the vancomycin was about 750 micromolar you need, right? So here in the case of dipivan, you need about only 1.2 micromolar. So it's about 700-fold more active than vancomycin. Just a single change, just like a small change with a rational design, we can get the activity back. And further, so the bacteria face a real difficulty in acquiring resistance to this compound because of having a new mode of action to the dipivan, right? And then here, to check our hypothesis, so we check that lipid pyrophosphates, and then we added the dipivan, the physical mixture, in presence of zinc 2 plus, and then we check the formation of that complex by mass spectrometry, and we see that so this compound makes the this compound along with the zinc 2 plus complex makes a complex with the pyrophosphate, and then inhibiting the cell biosynthesis. So and then further here, so we wanted to check if we add the pyrophosphates to our assays, to our activity assays. Say suppose you are providing the target in the assay. That means the compound is being nullified by the external target. So when we add that externally, then we should not see activity. That's exactly what we see, saying that so this compound, like these pyrophosphates, is acting as an antagonist to this dipivan, indicating that this molecule is actually binding to the pyrophosphate and then acting on the bacteria. And then we checked the activity of this dipivan in the kidney infection model, and then as expected in the in vivo, so this compound showed a very good activity in the kidney infection model when compared with the vancomycin and the linezolid. So with that, so I would like to move on to a second approach that I was applying in my postdoctoral research. So where I was working on the natural product drug discovery. So so basically in the previous slide, I have shown you that most of our antibiotics actually come from the nature. So that natural antibiotics actually mostly have come from a bacteria called Actinomycetes. So that Actinomycetes bacteria, they are rich in the antimicrobial producers. And then, so during my postdoc, I was applying a not targeted activity approach of antibiotic drug discovery. So basically, by doing this exercise, we can find novel antibiotics, or we can uncover new activities for the old antibiotics, and also we can perform the synthetic biology or genomic engineering. So during my first project, we were working on the discovery of antibiotic adjuvants for immunoglycosides. So as I said, the adjuvant is not active by its own, but when you have in combination with an antibiotic, it should have activity. So we were identifying the extracts to be active in combination with gentamicin. So basically, the gentamicin is an aminoglycoside antibiotic that inhibits the protein biosynthesis in bacteria. And further, so bacteria acquired resistance to gentamicin, as I said in the previous slides, by modifying their target sites, or by modifying the antibiotic, or by efflux pumps. So here in my postdoc lab, we have so basically what we do is, so we collect the soils from different environmental sources, and then after collecting the soils, we specifically treat the soil with some of the antifungals and some gram-negative antibiotics, and then isolate specifically actinomyces bacteria. And then so those bacteria we isolate, and then we make the extracts, small-scale extracts of those bacteria. And then once we make those small-scale extracts with some suitable organic solvent like methanol or ethyl acetate or DMSO, so once we make those extracts, we check for the activities. And then I took a bunch of extracts to see the activity against the drug-resistant MRSA that carry an enzyme called AAC-APH enzyme. So basically, so we were here looking for the extracts that potentiate the activity of gentamicin, right? So here we have identified a few extracts that do not have activity on their own, but when you have combination with antibiotic like gentamicin, that has the activity. But the one that I highlighted here, the VAC9126, which we call the extract name, so that extract having the reproducible and a very good activity. And then so here I show you a disk diffusion assay. So basically on the disk, so the black circle here that is showing here is a disk, so where we load the extract. And then if you see there is no growth, if the extract is showing any activity, then you should not see the growth of the bacteria around the disk. So that's what we see. Like in the case of VAC9126, so it is not active on their own. So that's why it is it doesn't show any inhibition of the growth. But in the case of gentamicin, it showed like slight inhibition of the growth around the disk. And in the case of VAC9126 plus gentamicin, when you have in combination with the extract, you see like a big halo, a big zone of inhibition, indicating that the extract with gentamicin showed a very good activity. And then so now we identify the extract that having the activity initially, and then once we identify the extract, so we need to find the compound, we need to identify the compound from this extract. So basically to identify the compound, what we do is, so we grow the culture in a large scale, like from the 3 liters to 10 liters large scale, and then for about a week we grow them in an incubator. And once we grow them in an incubator, we extract those bacterial cells with a suitable solvent like methanol or ethyl acetate. So to get a crude extract, something looks like this. Like sometimes it is worse than this. So once we get the crude extract, we do a series of purifications to identify the active compound. So basically, during all these purifications, we do activity. So we do a bioactivity guided purification. So we check the activity at each and every stage of the purification to see if activity is carrying over. And once we purify the active compound to have something like this kind of pure peak, and then we do a series of studies, like character studies, to identify the compound, to identify the structure of the compound. So by doing to check the purity by HPLC, and then high resolution mass spectrometry and then MSMS, and then 1D NMR, 2D NMRs like COSY, HSQC, HMBC, sometimes noisy, and even if those are even not enough to get the structure, so what we also do is we do degradation studies. And we also do sometimes, if we don't even get the structure by doing all these studies, we do synthetic biology manipulation, like we edit the genes in the bacteria and then to identify the structure. So from this extract, like from after doing all this exercise from the VAC9126 extract, so we identify the compound called venturicide A. So this compound is the active compound. So basically, like after purifying, we checked the activity of this compound in combination with gentamicin. So in a checkerboard assays. So basically here on the x-axis we have the gentamicin, on the y-axis we have the venturicide. So if you see the color dark color, that means that means the bacteria is more. So that means there is no activity of the compound. So in the case of gentamicin, you need about 64 micrograms per ml to kill this bacteria. Where in the case of venturicide, it was not active on its own, as I showed you in the previous slide. So, but when in combination of these two compounds, you see the sweet spot at 4. So the 4 of venturicide and the 4 of gentamicin, you see the activity here, which is a sweet spot, which is so in combination, this is working with a pure molecule. and then the same pattern was seen with the Enterococcus faecium. So and also we see with the other bacteria like Pseudomonas aeruginosa and Acinetobacter baumannii. And once we identify the activity in vitro, and then we wanted to check its mechanistic investigation, like how this compound is helping gentamicin to kill the bacteria. So basically, I would not go into the details of it. I would just tell you the summary, like what we identify here. So basically, what this venturcidin, what it does is, so it inhibits the ATP synthesis in bacteria. So you all know that like ATP is the powerhouse of the cell, right? So we need ATP, or any cell or any organism, they need ATP for survival, right? So what venturcidin does is, so it inhibits the ATP synthesis. So by blocking the proton translocation, so this proton translocation is necessary to generate the proton motive force to make the ATP. So basically, what venturcidin does is it blocks the proton translocation, and as a result, you see the more accumulation of protons on the outer surface, right? So if it is, so we thought that maybe those protons, whatever is accumulated on the outer surface, may be linked to the gentamicin potentiation. So to check that hypothesis, so we used a proton of 4, a compound called CCCP. So this is a proton of 4. So basically, what it does is it takes up those protons, right? So when it takes up those protons, if those protons are involved in gentamicin potentiation, then we should not see the potentiation activity here when we add the CCCP in our assays. That's what exactly we see that. So CCCP nullifies the venturcidin effect. So it completely antagonizes, indicating that those extracellular protons are linked to the gentamicin potentiation. And then further, so this venturcidin, is helping gentamicin to go into the cell more efficiently than not having it. So to summarize the mechanism of venturcidin, like how it is potentiating gentamicin, so basically, as I mentioned in the previous slide, so it blocks the proton translocation and then inhibits the ATP synthesis. So as a result, you see the more accumulation of protons on the outer surface. And then because of those protons, which are acidic, right? So now it becomes acidic region. And now if you see the structure, if you remember the structure of immunoglycosides, so those immunoglycosides, they carry at least 4 amino groups, primary amino groups or secondary amino groups, which are basic in nature, right? So because of it is just now acid-base interactions. So because of the acid-base interactions, those protons, those amino groups on the immunoglycoside molecule, so it becomes cationic, and then they're being engulfed by the bacteria, and that is how this venturcidin is helping the bacteria to be uptaken more, and then killing the bacteria more efficiently. And then so after identifying the venturcidin from the VAC 9126, so we wanted to identify its biocentric pathway, right? So I showed you the venturcidin structure, right? It's a very big molecule. So to make that molecule chemically, we need lots of reagents, lot of synthetic steps, right? So and we need a lot of effort. So how does the bacteria make that molecule within their cells, right? So to identify the biosynthetic pathway, so what we do, so all that pathway codes are the information is actually is found in the genome. So to identify the genomic pathway, what we do is like we extract the whole genome from the culture. So we grow the pure culture of the bacteria. We take the pure culture and then we extract the whole genomic DNA from that culture, and then that DNA, so we sequence them. So you all know the like deoxyribonucleic acid, right? So it contains like base pairs like adenine, thymine, guanosine, and cytosine. So they are like combinations and permutations, right? So basically, we read those ATGC sequence of those DNA that we isolated from the culture, and then after reading that those sequences by sequencing machine, so we assemble those reads by bioinformatic analysis, and then we perform an anti-smash analysis to predict the gene cluster, like that can probably is making this molecule. We predict the cluster initially by bioinformatic analysis. Once we predict the cluster, so we predicted the cluster from VAC 9126 producer like to be a 94 kilobase pair. So all these arrows represent each gene. and each gene makes in each enzyme, and each enzyme makes each reaction in the pathway, right? So these are all the genes we predicted to be involved in making this molecule. So to confirm, this is the gene cluster. These are the genes that are necessary for making this molecule in that producer. So to confirm that, what we do is like we transform, like we clone that, we take the, so we capture that gene fragment from the producer, right? So we capture that, and after capturing from the producer, so we transform that into a vector, and after transforming them into a vector, and then we put into the host called Streptomyces silicolor 1146 and 54. So this bacteria originally does not produce, but once we add the gene cluster that we predicted, then this surrogate host should produce the ventricular molecule. And then so after having the successful transformation, so we checked by HPLC and NMR to see whether these clones are producing ventricidin molecule or not. So indeed, so the cluster that we predicted so is actually responsible for making ventricidin in the producer. So here, so the one that VAC 9126, so that I showed the HPLC in the red color, is from the producer strain, the original producer, and then these are the clones, silicolor 54 and 46, which are the clones which originally they do not produce, but after adding the gene fragment that we predicted, so after adding the gene fragment, we see the bacteria now producing the ventricidin, indicating that what we predicted is actually making the ventricidin molecule. Then I would like to move on to the second strategy that we developed by doing genomic mining. So basically, I said in the previous slides, most of our antibiotics discovered from the bacterial sources. I just said also Actinomyces bacteria, and I also said that the antibiotics kill bacteria, right? So now here I ask a question like, how come the producers of antibiotic do not kill themselves, right? So they must be having some self-resistant mechanism to protect themselves from the antibiotic production, right? So here we screen for the presence of such antibiotic resistance genes in the producers by genomic mining. So we use that self-resistance mechanism to identify the antibiotic producers. So for doing that, so we choose so to check our hypothesis, we choose an antibiotic called alpha-mycins. So these alpha-mycins basically they inhibit the so they bind to elongation factor 2 protein in the protein biosynthesis. So basically they inhibit the protein biosynthesis. So I would not go into the details of the mechanism here. So they inhibit the protein biosynthesis. So basically these molecules, they bind to elongation factor 2, and bacteria acquire resistance to these molecules by modifying their mutations. And then so the mutation that having the alanine 375 309 having the maximal resistance to these kind of molecules. So now we wanted to screen for the presence of this particular mutation in bacteria, particularly in Streptomyces bacteria, to screen for the producers. So we screened about 2,000 whole genomes for the identification of this A375T mutation, and we have identified about 21 Streptomyces species to have this mutation. And then we also predicted that maybe that mutation maybe is associated with the gene cluster. So with that criteria, we have so from this 21 so initially 2,000 species and now come to 21, and then with this criteria now it come to 3 species, only 3 bacteria. So in the 3 bacteria were found to be like Streptomyces cattleya and sulfurious and NBRC 110611. So the cattleya is known to it is known in the literature to produce a molecule called L681217, which is a chiromycin class of antibiotic. So indicating that is an alpha-mycin. So and these two bacteria are not known to produce this kind of molecules. So here I show like each arrow represent a gene, and then the whole genes are regulated by the cluster. It's called gene cluster. So they all carry the chiromycin resistant elongation factor 2 protein within the cluster. So as I said, the Cattleya produces a known alphamycin, and then these two, we don't know what it produces. So we have purchased the sulfurous strain, and then we have performed the complete extraction the procedure to identify the active compound from this strain, so from the sulfurous strain. So we have identified the compound from this sulfurous strain to be phenylpharmacin A and B. So this compound, as you see here, so here we added EF2 protein within the assay when we do it. So basically EF2 as an antagonist, so since this compound binds to the elongation factor 2, so we did not see activity, indicating that these compounds are binding to the elongation factor 2 protein, indicating that these are alphamycins. Once we as I said, so we also identify the biosynthetic pathway of this compound in the producer strain. So we identify the genes, and then that make the enzymes to catalyze each and every step during the synthesis of this molecule, and then we confirm this pathway by doing the heterologous transformation, by doing the cloning experiment, like we did with ventricidin, to confirm the pathway of this biosynthesis. And then further, we checked, once we get the pure compound of the phenylpharmacin, so we checked the activity of this compound against the drug-resistant Neisseria gonorrhoeae. So basically, here I would like to stress that this gonococci bacteria are gram-negative bacteria. So basically, right now at this present time, so we only have one treatment regimen to treat this infection. So we are now lack of effective therapies to treat these infections. Now we see that the compound that we isolated from this sulfurous strain, so is having a very good activity against multi-drug resistant Neisseria gonorrhoeae. So here we compared its activity against the other drugs, with other drugs like erythromycin, azithromycin, tetracycline, gentamicin, ciprofloxacin, cefotriaxone. So when we compare with these drugs, this molecule showed a very good activity, phenylpharmacin B. So and then as I said, these alphamycin molecules, they inhibit the protein biosynthesis, right? So to check to see whether this compound is inhibiting the protein biosynthesis or not, so we did the in vitro transcription translation assay. So basically, so there is a luciferase enzyme. So the luciferase enzyme synthesis. So basically, we take a cell extract, and then with the cell extract, we have certain genes in them. So with that genes, so we provide the antibiotic with the assay, and then we measure the synthesis of a protein. So here, if we see, so in the case of tetracycline phenylpharmacin B, so we see that the protein biosynthesis is being inhibited. And then further phenylpharmacin binds to EF2. So here this is the native page assay. So basically we run the protein on a gel. So basically here is the protein ladder. So and then here in the case of 2 and 3, it's just the EF2 protein. It does not have the antibiotic here. So you see now in the case of 2 and 3 rows. So you see that the protein is like is not compact. It's a kind of broad, and then in the case of 4 and 5, you see where the antibiotic is there. So you see the protein has become compact. So indicating that this compound is binding to the is binding to the protein. So what this compound does is, so we identify in the crystal structure. So what this compound does is this phenylpharmacin compound. So it binds. So this protein has 3 domains: domain 1, 2, and 3 domains, and then this compound binds to in between domain 1 and domain 3. And then closes and then it compacts the conformational changes, and then closes the conformation to make the compact shape. And then and that is how these compounds inhibit the protein biosynthesis. Okay, so with that, I would like to I hope I have shown you today some of the infectious disease topics, and then antibiotics and their importance, and then bacterial resistance to antibiotics, and then I've showed you some of the strategies that we developed during the past decade in my research. And then here I would like to end my talk with this slide. So as I said, like we do not have the antibiotics right now at this time point. So for example, imagine the COVID-19, right? So if you see our COVID-19 regimens, the treatment regimens, you might be seeing that So we have like azithromycin. People like the doctors prescribe azithromycin for almost every patient, right? So indicating that you might be developing a co-infection with COVID-19. So imagine if we do not have azithromycin, if we do not have those antibiotics at this time point, then the death rate might have been even more devastating than now, right? So we need to protect our antibiotics. We need to have the right diagnosis to take the right antimicrobial at the right time, at the right dose, through the right route, and for the right duration. And also we need to have the right documentation to protect our antibiotics, and then for our future generations. So with that, I would like to thank the PB Siddhartha College Department of Chemistry for giving this opportunity. And I would like to thank my PhD supervisor, Dr. Jayanta Haldar, and postdoc supervisor, Dr. Jerry Wright, and the colleagues in both the labs, funding agencies like JNCSR, CSIR, and McMash University, and other centers. And I would like to thank you all for your kind attention. I would like to have, I'll be happy to take any questions you may have. Thank you all.
Thank you very much, Dr. Venkateswarlu Yarlagadda. It was indeed a very great seminar. You have combined your research experience with this topic in an excellent manner. And now I request all the participants to post your queries in the chat room. If you have any queries, I request the participants to post them in the chat room so that our speaker can address the questions. If you do not have any questions, we will proceed further with the vote of thanks. Now I request Dr. TNVSS Sachadev to give the formal vote of thanks.
Hello, a very good morning to one and all. It gives me immense pleasure to propose formal vote of thanks at the end of this international webinar. I'm extremely thankful to the resource person, Dr. Y. Venkateswarlu, for accepting our invitation and making this webinar a grand success. Venkateswarlu, once again, thank you for accepting this and making this a grand occasion for us. I would like to propose my sincere thanks to the management of PB Siddhartha College of Arts and Science for supporting us in all our endeavors. I extend my sincere thanks to Dr. Ramesh Garu, Principal PV Siddhartha College of Arts and Science, for supporting us always to organize such programs. I would like to convey my heartfelt thanks to Professor Rajesh C. Jampala, Dean, PB Siddhartha College of Arts and Science, for giving us valuable suggestions and supporting the department in organizing such activities always. I also extend my honest thanks to Sri V. Babu Rao Garu, Director, PB Siddhartha College of Arts and Science, for encouraging us to organize webinars. I am also happy to extend my thanks to all the colleagues of my department for their committed support in making this endeavor a successful one. Last but not the least, I would like to convey my sincere thanks to all the participants in making this webinar a grand success. Thank you all. Now I am going to post the feedback link. Please fill the link so that you can receive your certificates.
Oh, dear participants, Dr. Alice Jayaprada, you can post your doubt in the chat room, please. Feedback link has been posted in the chat room. I request all the participants to fill the feedback link. I request all the participants to fill the feedback link. Meanwhile, I request Dr. Alice Jayaprada to please post your query in the chat room. Dr. Venkateswarlu Yarlagada, I hope you can see the query. Antibiotic resistance, is it a hereditary factor or is it an individual one? Hello. Yeah, there is one query for you, Venkateswarlu. Antibiotic resistance. Yeah, just.
Yeah, I'm not. Yeah, yeah, yeah.
Antibiotic resistance, is it hereditary or is it only an individual one?
So it is antibiotic resistance. It is not hereditary. So basically, so the antibiotic resistance is like, as I said, like it's like acquired resistance by the bacteria. So when you treat with the antibiotic for a long time, so bacteria withstand the pressure of antibiotic, and then slowly develop resistance to the antibiotic, like with the serially, and so it is a genetic material basically. So initially they develop certain genes to make the resistance, and then the genes transfer between the species. So it is not like hereditary in our bodies, but it is probably hereditary for the bacteria. Did I answer your question?
Yeah. Any more queries or questions? If there are no questions, we can close this webinar. So I request all of you once again to fill the feedback link for getting the certificate. Thank you, Venkateshwarlu.
Thanks, man. Yeah.
Feedback link రాలేదా?